Very sorry for this technical.
Just a second. It's coming again. Suddenly it dropped actually. I don't know what happened.
Yeah, just stop it.
No problem. So I hope the slides are visible. Yeah.
Yes, yes, it is visible.
So anyway, no problem. So this was what I was trying to explain. That's my lab basically. And so these are the areas. So what I said that sensors is not a basic brand, it's applied branch. For example, the soil has various type of chemicals, metal ions, this, that, so many things. So that can be tested using a conventional method of HPLC, many other methods. So even the heavy metal ions or any metal ions, any kind of things. So that can be tested through sensors also. So that is important. For example, the basic sensor what everybody is looking in the lab is a pH meter. It's nothing. It's ion selective electrodes, basically the pH meters. Moreover, if we talk about the pathogens, basically, so pathogens is not limited in animals or human beings. It is very much present in the agriculture field. A lot of fungus, a lot of bacteria, a lot of viruses. We are showing a lot of examples. So I don't want to jump straight into the complex things because I know those who work in this area, they can immediately understand what is happening. But those who are new to this field, they should know what from where to start basically. That is my basic agenda of today's meeting basically. So anyway, so in this case, so first point, what we do, what we study in my lab. So so the first is a material engineering. Without material, we cannot go for development of sensor systems. Another is the commercially viable sensor prototypes. This one, what I'll show, we have developed 3 to 4 of that segments. Another is a disease diagnostic. Disease means what? I'll be showing in the next slides how we define a disease in a plant or animal or anywhere. Another is a nano engineering, which is very, very crucial and very important because the way we talk about the nanostructure, it is highly tunable things, right? Tunable means you change the structures, the physiochemical, magnetic, and every property will be changed. So that's the key basically. That is why we call it as nano engineering. And after that, so it's an electrochemical biosensor. That's one of the electrochemical system which is very important. And another is a microfluidics paper-based biosensor. That all these areas what we work in my lab. All right, so these are few examples where you will see that by what this all data belongs to my lab. So one is the aspect is the nanomedicine what we do. Another is one of the example paper based microfluidic system. You can see this is our recently filed patent. This one. Another is a biomolecular analysis. You can see these proteins in between because protein can be of plant origin, animal origin, human origin, any origin. Because protein is protein. So if we know how to make a sensor, that can be accordingly engineered, right? Another part what we study, as I said, these are the materials. These are the bimetallic system. These are the nanorattles. We call it as a damru in Hindi. These are the bimetallic system. Again, this is tree. This is outstanding structures. That's basically the tree-shaped structures like this. I'll be showing few of the examples. So we have a primary branch, secondary branch, tertiary branch, quaternary branch. So that we can develop in my lab. So that we have already developed and published a lot of papers in this area. Another is a nanosheet. So why I'm showing these things? This is important because these material is the basis of development of the sensor. So sensors cannot be fabricated in air. We need something, a solid surface, and onto the solid surface the matrix is needed. Matrix means some chemicals. So these are the chemicals what I've shown here in this side. So that can be utilized to develop these things. So another example is what we have done is ascorbic acid. Not only ascorbic acid, many drugs. Right, we have developed a lot of sensor. In this case, I have shown the mouse model. Mouse is little more typical. That can be done in the plant as well. So Basically, if I talk about the nano sensors in brief, basically, sorry. So what we study basically, we study the sensing, and sensing is nothing in general, more general term, it's a detection part. So what we detect, so we detect a COVID-19 antigen nowadays. Very much, everybody's talking so much about COVID-19, and then that's a whole cell. You can see the example: Escherichia coli, UTI infection, Aspergillus niger, Aspergillus flavus, Aspergillus fumigatus. Every type of the species and genus what I've said, they appear in plant system. You can obtain those from the plants. Plant pathogens, any kind of plant pathogen is very much possible using these sensors. The toxins, aflatoxin B1, any other toxin, DNA or gene, basically RNA and protein levels. So basically what I'm trying to explain, so when we go for a detection system, we need a certain molecules, and these molecules collectively is called as a biomarker. And when we detect a biomarker, then we detect a disease. So whenever we talk about a sensor, we always talk about detection. There is no other business. What is sensor and what will be the output? Output is detection. There is nothing else. It's a bioanalytical chemistry basically. And then there are two concepts basically. One is detect the antigen that are present in a diseased person or a plant, or else detect a chemical or a biological agent which causes a disease either in human or in plants. So my point is what? If you feel that this is a plant and this chemical can cause infection in plant, you detect this particular chemical in that matrix, or else in order to prove that this plant is infected, you just detect the pathogen or a chemical or any other oxidative stress event that is possible by using the sensor system. So one is which may cause and another is which has already caused. So this helps in designing the sensor system. That's a very important take-home message from these 2 points. So anyway, so the point is what? These are the very powerful systems. You can see here ELISA is there, HPLC and so on, PCR. So these are very powerful system. It is extremely powerful, but the major issues with these conventional method is what I never said that the conventional method is not good. They are very good, but still there is always a possibility to improve the system. So how to improve? What are the points which brings the upper hand compared to the conventional system for sensors? That's a laborious. Conventional one, they require large handling volumes, time consuming, complex, trained personnel, lack of miniaturization, no on-site analysis. Now imagine you are going from Himachal to Kanyakumari to have a soil health. You cannot take this HPLC, right? You cannot take that. You need something very small, tiny device that you keep in your pocket, and then you can give the on-site analysis in Kanyakumari or any other place. So that is the key point for sensor system. So major challenges in case of diagnostics is quick detection, ultra sensitive detection, and on-site detection is the major upper hand for the sensor systems. Okay. So the answer to these things is what? Biosensors basically. So biosensor is an analytical device that utilizes a specific biochemical reaction mediated through enzymes, immunosystem, oligonucleotide, tissues, organelles, or whole cell. So it means what? There are two things. There are two components basically. One is the receptors, another is a transducer. In case of the receptor is antibodies, either a peptide, aptamer, enzyme, DNA, RNA, and so on. So these are the molecule. It's like hand. It will hold the compound, and then there will be internal processing. So this interaction of holding the compound is a biological event, but this biological event is not a measurable thing. So in order to measure that, you need to have a convertible readouts. So that here comes the importance of the transducers. They convert the biological event into a readable system. So if you see the next slide here, that transducer which was blue here becomes red. And here the signal output is coming. So what happened? These molecules, this molecule, they interacted with this one or this one or this one or this one. They don't come together. This or this or this or this or this or this. So they interact and there is a biological event. The transducer converted this event into the measurable signals. So eventually again there are two things. One is the receptor, another is the transducers. All right. So As I said earlier, there is a wave basically. You can see this market report 2021 July 1st. So you can see the transparency market report, what they say basically the medical sensor market or any sensor market, they have a very high importance in terms of the business basically. So in this case, you can see there's a real-time monitoring, ultra-sensitive detection, non-polar detection, and ability of miniaturization. These are the key points which is very much important. And not only this one, you can see the allied market research also says these kind of things. So eventually the point is what the sensor system is very important, not only in agriculture, but in every field where there is a target molecule. Okay. So, so eventually the analytical work in the future, how it looks like, everybody who is sitting here, I'm sure my friends, they know the PCR basically. Okay, they know some kind of separation, HPLC or any kind of thing. So this thing can be tuned onto the small chip. So person 1, person 2, person 3, person 4, person 5. So all this together can be trimmed onto small chip. So that is what it is called a lab on a chip. So for example, separation, concentration, detection, and final analysis can be done on a single chip. Even I have done it. So this thing gives you the lab on a chip system. So that's the future of the detection of any molecule which is sitting in any matrix. So I'm sure that my voice is going
Yes, it is sounding clear.
okay because there is. I mean, we can have a discussion in between also. No problem. If there is a doubt in between, anybody can ask. I'm okay with that. All right. So if you start studying the sensor, the most important are these boxes. Without these boxes, we cannot even imagine that how the sensor can be fabricated. First is what to detect. When I say what to detect, it's what a small molecule. Another is what a macromolecule. In macromolecule, that could be many things: a DNA, a protein, sorry, a DNA, RNA, protein. So depending upon the type of the target, the fabrication strategy changes. And when we know that we are going to detect a protein, so we'll design the sensor accordingly. When we are going to detect an antibiotic or a toxin, aflatoxin or ochratoxin or any other toxin, so the strategy is different. Okay, so molecule, what is the physiochemical property of the molecule? So depending upon what we design a sensor, and when we design a sensor, we think rationally that okay, so if the protein is this, so the surface will be insulating. If the molecule is conducting, the surface should be like this. So when we hypothesize something, then we make a scheme based on the thought. So that's a thought process, right? A scientific thought comes here, a scientific rationale. That's a scheme basically. If you see any paper throughout the world of a sensor, they in 99% case they show a pictorial schematics. So that comes the thought process, and whatever you think that has to be fabricated, here you have to dip hands. It means you have to do it here, and here you have to think, but here you have to do. So just a second. All right. So in this case and in this case, we have to think, and in this case we have to do. Fabrication means you have a surface, you have to do layer 1, layer 2, layer 3, layer 4, and so on. So when you have done this layering, it means you are coating some chemicals, chemical 1, chemical 2, chemical 3, and so on. So you are doing the experiment here, but whether it has been done correctly or not, this box will tell. So this is an order of the experiment that has to be done without any exception. That has to be done. Otherwise, we'll not know whether the sensor is fabricated correctly or not. So we have to characterize. Characterize means what? Any characterize, physical, chemical, electronic, anything. So there are
Excuse me, sir.
numerous techniques. Sorry.
Excuse me, sir. I have
Yeah.
a doubt in the
Yeah.
fabrication.
Please go ahead.
Sir, whether a nano sensor can fabricate in India? or we have to depend on other
Can you speak a
countries
little loud?
for.
I don't know who's speaking. Ajit, Mr. Ajit,
I'm,
and
I'm Ajit. Yes, I'm Ajit Kumar here. I
yeah,
just
please.
want to know fabrication can done here in India itself for nano sensor fabrication.
Mhm.
Fabrication process we can do here in
Yeah, of course.
India.
Yeah, of
Labs.
course. Absolutely no
Okay.
problem. You can do it in my
Oh,
lab.
okay. Okay. Thank you so much, sir.
No
Okay.
problem. So there are many places in, I mean, most of the IITs they're doing that. I see they're doing that. No problem. You can visit my lab. I will show you how to do that. So anyway, so where I was, yeah. So fabrication, I mean, of course, if you cannot fabricate, we can't make a sensors, right? So the characterization means physical, chemical, electronic, whatever you can think of instrument in the central instrumentation facility anywhere in IIT Delhi, IIT Varanasi, Kanpur, anywhere. Every instrument can be helpful in giving you the output. Characterization means in Hindi, if I'll say, it means when I teach in the class, I say sahi bana hai ki nahi bana hai. It means the sensor has been correctly designed or not. This data will tell you, and you will get at least 100 type of data here, or maybe 10 type, 20 type, depending upon how vigorous you are interested to get the data. Anyway, so after this fabrication process and characterization process, we'll go for the optimization. Optimization required, there are two types of optimization. One is related to fabrication and another is related to what? The data output. So why I am saying these all boxes? Because whatever I am going to talk after these, fundamental aspects, every data will have these things, most of them. So you need to be very sure that what is the meaning of these, components. So fabrication optimization and data output optimization. I'll give you one example with reference to fabrication optimization is what. If we went in a party, we have, for example, 6 seats, chairs. And if we mimic these seats with antibodies, I'm sure everybody know, Eliza. If there are 6 antibodies, then you will have a limited number of antigen which can bind here, right? And if the limited antigen is not binding, then you still have a scope of increasing the signal. It is saturated. It means what? The binding site is occupied. So that is what is the meaning of fabrication optimization. Similarly, we'll go with the data output. It has many internal features. That is too detailed, but I'll tell you temperature optimization, pH optimization, molarity, and so on. Sorry. There are many things that has to be optimized here, of course. So after this optimization process, we will move on to the analytical performance. We'll move to the analytical performance. Right. We'll move to analytical performance. It means what? I'm just drawing it in a corner. It means what? The calibration. The meaning of analytical performance is calibration. I'll just talk numerically after this slide. What is the meaning of that? And then the analytical performance means whether the sensor design is able to detect a fusarium in the plant, whether the sensor design is able to detect aspergillus spores, whether the sensor developed is able to detect the components in the soil. So the meaning is this one. So after this process, so these thing happens in a lab. After this process, we go to the real sample analysis. It means what? A plant leaf, a soil, root, anywhere, even in the live plant in vivo model. Okay, so that's the basic thing. If your sensor is very powerful to detect the spores outside, but is not able to detect in the plant itself, it is not that good. So that's what is the meaning of the real sample. So after this process, there is an interference studies. What is the meaning of interference? The meaning is what? If it is going to detect A, it should not detect A dash. It should not detect B, it should not detect C. If they are detecting this one, the sensor is not stable. Sorry, it is not selective. That's the meaning. And the last is the stability. So stability can be of many times, the electrode to electrode stability, long-term stability. Why it is important? Because the shelf life is very important. If you are transferring the sensor from Himachal to Punjab or Himachal to Assam, if it is not stable, the basic fundamental need of everything has been lost. So that is very, very important when we talk about the sensor design. So these are the key points that have to be considered when we are going for the designing of any sensors. So this is a phase one of my talk today. As Dr. Arvind has said, if there is any doubt, you can please discuss with me. Otherwise, I'll move on to another section. Please tell me. Yeah.
I think it will be good if we can have discussion at the end of session, because otherwise we are actually running short of time. We have to
So I
complete.
hope that I've been given 2 hours, right?
Yeah, you have to complete 2 hours.
No, I can complete 5 hours. That's not a problem.
Okay,
But
then it's not an issue. It is up to
so
the participant. Like if they have any query, they can come
well,
up
that's
with
what
them.
I mean. We need to discuss, no problem, because I'm sure that is a new area for the participants.
Yes,
For
definitely.
me, I can keep on talking for several hours. That's not a problem. But if there
Yes.
is anything in between, people can ask me. I will be very happy to answer. All right. So I think there is no question, so I'll move ahead. Okay. So as I said, characterization, there are the various techniques in the labs. If you go to any CSIR lab or IITs, you might have seen FTIR, XRD, EDX, all these materials can be characterized using these techniques. These are the few, figures of these instruments basically. Again, so when we characterize those things, the meaning of analytical performance is what? Calibration. Calibrate. I'm sure everybody know ELISA. ELISA में जो आप करते हो तो उसमें एक straight line आती है. Y axis पे absorbance रहता है, and on x axis you have a concentration. That is calibration basically. इसका मतलब है analytical performance, right? Not technique, performance. So it means you are increasing the concentration and the signal is increasing. So that's the basis of even ELISA does like this. That's the basically optical readouts. So you need to do the surface characterization, you need to do the electro optimization, and then there are various techniques that has to be done before going into the real metrics. So there are some detailed mathematical equations. I will not go in detail, but there are one calibration system. Sorry.
Sir, I have a solution.
Yeah, please.
Hello.
Yeah,
Sir,
please.
just want to know how we can identify the purity of the nanomaterial. If there are impurities there in the
Yeah, of course,
nanomaterial,
of course. Yeah, of course, of
how
course.
you can identify?
See, if you, for example, you have prepared graphene, for example, right?
Okay.
So when you do the elemental analysis, right? For example, if it has the impurity of lead, right? So when you do the elemental mapping or analysis, you can get a signal of lead. Or
Okay.
else, if you are doing the EDX, right?
Okay.
Then you will get a impurity. Or else, I've shown it here. These techniques: XRD, EDX, mapping,
Extra effect.
estimating. So you will be knowing by these techniques. No problem. The
Okay. Okay.
purity of the
Thank you.
material can be, confirmed by these techniques. Okay.
Okay, sir. Thank you. Thank you.
All right. So these are little more detail. I'm not going into this one, but I'll talk this part a little more. The next slide, most likely. Yeah. Okay. So basically, this is analyte concentration. Please skip this part. No need of this. So this is the analyte concentration. Analyte means target. Okay. The target can be, I said, small molecule. It could be a DNA, it could be RNA, it could be a protein, it could be even gases, it could be a free radical, anything, anything. When you're increasing the concentration, the signal is keep on increasing. That is the basic output of any sensor. But there are various points in between which talks about the LOD, it's a limit of detection. This is the sensitivity, is the slope. Sorry, this is a limit of quantification. I'm sorry, this is a limit of quantification. This is DL is a limit of detection. So how it works in a sample system, I'll tell you in the next slides. This one. So this is a with reference to this is a glucose example, but this can be extrapolated for any target molecule, any target molecule. So basically, what happens in a sensor, everything is a level. So as I said earlier, also the sensor give us the information with reference to the amount of the target present in a given sample. The sensor does only this job, nothing else. Okay, so for example, if I talk about the low glucose is the hypoglycemia, high glucose is the hyperglycemia. The range is what? From 50 mg per dl to 500 mg per dl, for example. So if this range is wide, it means if the blood, if the glucose is present 100, 200, 300, 400 and so on, the sensor is able to detect every level. But if it is very narrow, for example 50 to 150, this is a very narrow range. So that is what the meaning here comes, the dynamic range, and this is universal truth, and it is applicable for every type of sensors throughout the whole world without any exception. This dynamic range, the meaning is what? Dynamic means what? It should be as wide as possible. So that is the main thing. Hello?
Yes.
Okay. So this is the meaning, dynamic range. It should be as wide as possible. So numerically, if you know, this is the limit of quantification, this is the limit of detection, this is the limit of linearity, and this gives the sensitivity of the sensor. So if you go in agriculture, if you go in human biology, if you go into plant, if you go into soil, if you go in air, if you go in water, if you go in jungle, if you go everywhere, you will get this plot without any doubt. So the performance of the sensor is governed by this particular diagram, this particular plot. So basically now there is one thing which we talk about the sensors and nanosensors. Biosensor means there is no incorporation of nanosensors, and nanosensor means it is not mandatory to have a biological matrix in that. For example, antibody or a DNA or a RNA or enzyme or any other aptamer or anything. But generally, we combine the bio nanosensor together to why? Why we use them? To conjugate. For example, this is the gold nanoparticle, this is some conducting polymer system, this is a nanocomposite, NC. I'm just writing like this, nanocomposite. So what is this giving? This is giving a functional group, and you can have a CONH bond here basically. If you have a CONH bond, that is a complexation of the antibody. This is antibody, this is a gold. So that's a bio nanosensor or nanobiosensor. Then is a target interaction. That's a conjugation of the receptor. As I said earlier, receptor can be anything. It could be antibody, it could be peptide, it could be anything. And the next is what? The miniaturization and non-enzymatic approach. This is one another approach. And another is what? The signal amplification. This is without nanomaterial, and this is with nanomaterial. You can see the signal is increased when you're incorporating the nanostructures in the biosensor design. So this is the scope of the sensors. What we have done in my lab, that you can see in the fermentation industry, in the biomedical analysis, environmental monitoring, you can see in one shot so many metal ions can be detected: lead, titanium, cadmium, zinc, copper, nickel, iron, and it is not in human blood, it is in the surface water which is moving into the soil. So that can be done basically by using this system. So the bacteria present in the surface water, food processing industry, pharmaceutical industry, drug analysis, and so on. So this is the scope of sensors. As I said in the starting of my presentation, that we can keep on going and going and going in any other matrix, any matrix. So I'll give few of the examples. So these are the basics thing what I wanted to take a little more longer time. So now I will be a little more faster to give you the example. So these terms now it will keep on coming, characterization, this and that. So again, I will giving the example of ALP in my case. So these are some the device which is present in the market with reference to sensor, not for agriculture, but of course for anything that's possible. If it can detect the COVID-19 antigen virus, so it can detect any plant virus, right? Because sensor, it is a prototype basically. Okay. So these are some examples where the paper diagnostic can be, I'll show you a few examples here recently, the commercialized sensor for agriculture field. Now, this is one of the sensor what we made. This is office punching machine. We punch the paper and we have a disc out of that. We have done some chemical analysis, chemical, reactions here. and then we have done the anchoring of this receptor. After this receptor, this is a protein which is the ALP. It is present in many matrices. So it will make a complexation here, and then this presence of ALP will do this particular reaction, whereas the BCIP is the substrate, and then there is a dimer is formed. So you will get a colored product here. So based on this RGB analysis, we can do the qualitative estimation by naked eyes. So that is the main sensor what we have fabricated in my lab. So what we thought that we can do a qualitative means yes or no, but quantitative means we need to know the exact concentration. So we linked this system with a mobile phone, what everybody have. That's called as a DIC. It's a digital image calorimetry analysis. Then we have a calibration system. I already explained this in the previous slide that this is the analytical performance. That's analytical performance. Okay. So the innovation and the translational potential is what in this case, basically it's a waste paper disk. Nobody used this disk after punching the Waterman paper in the lab. So that's a low cost affordable technology. You will get a yes no analysis. For qualitative essay, for quantitative, you can integrate the smartphone. So my point is what this kind of the tiny sensors can be made in the laboratory. Again, this is the characterization. I will not go in any detail of this. This is FTIR data basically, Fourier transformation infrared spectroscopy. When you are doing this, I said layer 1, layer 2, layer 3. The signal changes in this box. So that is what I'm trying to try. I said earlier also. When you load chemical 1, chemical 2, chemical 3, the layer changes. Now you see here, layer 1, layer 2, layer 3, layer 4. What is happening? This nanometer is increasing, you see. It means something is onto the surface, and this surface texture is changing, topology. That's we call as a topology. So basically, when you are adding, for example, a book here, one book, second book, third book, fourth book, the height is increasing of the pile, basically. So that's what is happening at the nanoscale even. So after this whole process, we moved on to characterization, also analysis in the milk directly. You can see in the milk, the red curves they keep on increasing, the black curves keep on increasing with the increasing in the concentration. What is if we plot it without histogram? It's a straight line like this. What is this? This is a signal and this is the concentration. What I already explained earlier also. So in order to confirm that, we have added this concentration in the milk and we have tested this one. So we tested this concentration and this is the recovery percentage. By this you can calculate by using this equation, you can calculate the recovery percentages. So we moved on to standard addition method. This is one more method. It's more complex method, but simply you can understand this is a raw milk basically. So you can know the raw, the concentration of the ALP in the raw milk by using this particular method. So we have done it and we have found it is 17.5 unit per ml. So that is one of the important findings. So this is final device what we have developed in my lab. You can see these are the dimension of the device and you can see the size. This is a 5 rupee coin. And you can see the size of this device. This is before and after. This is the portability of the device. You can see you can keep it in a wallet. You can see the color chart. By this color chart, the farmer can know that yes, the milk is pure or not. And you can see the weight of this device, 0.38 grams. So this is the first report of digital image calorimetry based detection system. And it takes 13 minutes to detect the whole thing. So we have a lot of news in this case. We also appeared in the Rajya Sabha TV. This was the 12 most innovative sensor in the whole world. This is one of them, and it was 1 lakh, 1.7 lakh views in just 1 hour, one or 2 hours. We have on that this one in the news, and FSSAI also endorse our research. So after having this system, because we said we will take from the mobile to because when you're taking the light, when you're taking the image from the mobile, the external light makes some problem. So we rectified those things by having the tunnel. We have developed this device recently in my lab in Varanasi, and we have already filed a patent for this. So this is a patent application number. And then you can see this device. You can integrate this sensor into that, and then you can simply operate the the smart board, and then you will get the readout signal. So that's how it happens basically using the system. So I'll not go in much detail, but I'm telling how this sensor system can be miniaturized and can be tested for on-site. The size of this is basically I can keep it on my palm, on my hand. So we got the Gandhi and Young Technology Innovation Award in 2020, and recently we also got the ICMR award for this particular invention. Okay, now I will move on. This is fun. I'm sure everybody who's sitting in this meeting, they know this particular structure. So in this case, what is happening? This is a defined pattern. So now if you've seen the ice, just put a hammer on that, you will get this kind of structure. This is called a snow formation, and this everybody knows. That's a neuronal cells. So we can make these structures onto the chip as well. What is happening in this case? There is a central branch, secondary and tertiary branches, or maybe even more. So this type of structures we have fabricated in my lab. It looks something like this: primary, secondary, and this one. So these are the nanostructures. So somebody asked me that how to fabricate. So that is very much possible in the lab, not in any lab who work in this area, right? So this type of nanostructure has been fabricated, and that it has been applied for detection of various molecules. So I'll just go a little more faster here. That's a DLA basically. It's a diffusion limited aggregation. This is the process of the formation of the dendrites. That is called as a dendrites. So in this case, what we have done, we have taken one small chip and we have fabricated the gold structures, and we went for the analysis of hydrogen peroxide through this reaction. Okay, so that's I'll skip. So under microscope, you can see these 3D structures. This is a horizontal view, and that's a cross-sectional view, right? Okay. Now, here you can see in the nanobiosensors, we have seen that a lot of terminologies we have used so far: fabrication, characterization, optimization, real sample, and so on. Now I'll give you few examples where the nanosensors can be utilized directly in the agriculture, plant, soil, any matrix. And what to detect? They have detected the DNA, they have detected the RNA, they have detected the gases, they have detected the heavy metal ions, and many more things. So you can see the application of nanosensors in agriculture. It's a biotoxin detection, and there is a hormone detection, it's a virus detection, herbicide detection, and bacteria detection. So I'll give you all the examples one, and then I'll explain how the data comes and how you will know that this can be utilized for a point of cure analysis, or how to know the soil health or the plant health. So basically, this is one of the examples. You see, this is a plant sample. This is an infected leaf. You prepare the sample sorry, by the detection of antibodies or by using the DNA system. So these are a conventional system like ELISA, everybody is doing that, or by the PCR. So these are the techniques what people are conventionally using to detect the particular target in a given plant sample. Now, this is basically the illustration of a green leaf volatiles. You can see jasmonic acid, salicylic acid, ethylene released during herbivory pathogen infection or abiotic stress. You can see that either the infection because of the pathogens or due to the abiotic stress, these chemicals they release. You can now imagine, for example, if this is a plant leaf and there is an infection, so the sensor, if you have planted here or somewhere here, somewhere near the plant, it should be what? It should be super fast. Like the pH meter give you the signal immediately, or the pH paper even changes the litmus paper. So one of the point here I would like to mention here, the sensor should be super fast. And as I said earlier also, you can go with the protein, you can go with the acids, you can go with the ethylene or anything. Like I'm sure that we know that people are using various type of gases, to preserve or for increasing the shelf life of the fruit and many things. So these things is very important when you are designing a sensor. Okay, now I'll give you one example of aflatoxin. It is a toxic metabolite. These are the chemical structures of that. Now the important thing is what? these groups, see, these are the carbonyl groups, right? This is the reference of this one. So these groups is what? It makes it electroactive. Electroactive means what? It can, for example, A is going to A plus and A plus is going back to A. That's a redox reaction. It means what? Reduction and oxidation or oxidation reduction. So these groups are capable to give you the electrons at a certain potential. I'm sure that people have seen the electron transport chain reaction. What happens on the crystal of the mitochondria? If you see the biochemistry book, they mention at several points that E or E naught, something like this. E naught. That's the formal potential of that. It means at a given potential is energy at which the electron generates. So that happens basically in these kind of the structures. So anyway, so my point is what in this case is what people are able to detect aflatoxin in various samples. These are the few general information. I'll not read it. So now let's see how it works. Now you see, this is the surface, okay, and this is one of the method of sonication. And you need to coat these things where onto the surface of what a glassy carbon. This is GCE, glassy carbon electrodes. Now you see, so these materials, gold, this, you have to coat it onto the surface of the electrodes. That's a glassy carbon electrode. It's like a pen-like structures. Now, after coating this one, you will put the antibodies here, and then this will have a conjugation reaction here. Conjugation reaction. And again, you will block this by the BSA here. You can see the structure. So if you imagine this part, basically, it is comparable to ELISA. I'm sure those who are from biological background, they know this word, ELISA basically. Now what you do, you put this aflatoxin B1, there will have a complexation reaction and the signal will come here. So I'll go back a little more again. So this sorry. So this is a chemical structure. It's not a protein, neither it's a DNA, and neither it's a peptide. It's a chemical structure. So this molecule can generate a signal, or else when there is a complexation here, this molecule will be selective and then it will generate a signal. You have seen here, there is a potential, as I said earlier also, and there is a current. What I explained earlier, A to A plus, that's a one electron transport reaction, transfer reaction. So this electron will generate what? Current. And that is on the y-axis here, on the current, and that is what it's a signal. So eventually what happened? Concentration and then the plot and the signal. So in this case, what happened? People have detected the aflatoxin B1 using a nanostructure surface where the gold nanoparticle was involved, and eventually the antibody was anchored, and then it made a complexation reaction and the signal was generated. So this is one of the examples. So before going into the data, I said earlier the word characterization, if everybody remember. So we have prepared some scaffolds here. So this is what? This is basically the scanning electron microscopy. This is what? That's a tunnel electron microscopy. So it means whatever you have prepared, this complex, that has to be characterized by various techniques. That's a scanning electron, there's a tunnel electron microscopy, and then this is electrochemical characterization. I'll not go in detail. This is a very complex phenomena. So that could be a very new thing for the participants. But these are the techniques by which you can know that the sensor is fabricated correctly or not. But in general, I'll tell you, you have a surface which is a metal, okay, and the electron passes through a metal. This everybody know. There is no, business of a sensor in this case. But if you load a protein, this, which is a insulator, and protein is an insulator basically, or it is a semiconductor. So the movement of electron will be increased or decreased. Can anybody can answer to this? Hello? Hello? So Mr. Ajit Kumar, my voice is going or not?
Going, sir. Good.
Okay. I asked one question, but I was expecting somebody will answer. But anyway, I will answer by myself. So anyway, so in this case, what is happening? There is really, if you putting an insulator or semiconductor, the electron flow through will be stopped. So that is what is the data here. Okay. So what is happening, guys? You are increasing the concentration of the target here. You see analytical performance, and what is coming? The concentration on the x-axis. On the y-axis, there is a current. So you are increasing the concentration onto the surface. There is a binding like this. This is the surface of the chip. Okay. When there is a binding, there is a drop in the current. When there is more binding, there is more drop in the current. What is happening? You see the drop in the current. With increase in the concentration from 0.05 nanogram per ml to 25 nanogram per ml, the current is decreasing. Okay, so that's what is the plot. According to this data, the plot is coming. So that's a storage time. That's a stability I already mentioned. And that is the selectivity. You can see this is aflatoxin B1, B2. This is G1, some other maybe ochratoxin or some other toxin maybe. So you can see the maximum signal is coming from this aflatoxin B1. So by this, sorry, hello. Hello.
Okay, I think the mic of someone, some participant was on actually.
So my voice is going on. That's what.
Yeah,
That's what
yeah,
I,
it is
okay.
clear.
So this one, what I'm trying to explain, this is a selectivity system. It means the sensor is able to detect only this aflatoxin. Others, either no signal or statistically very insignificant signal. So you need to do this in every case. Now again, that is spike concentration here and the detected concentration. So what is the point basically? 5 and 5.29, 25, 23, it is not comparable. So the recovery lies from 89, almost 90 to 105%. So this is basically what? This is basically in the spiked wheat sample. You see, wheat sample. Okay, so they have done this in the wheat sample. So it means what? The sensor is able to detect in the wheat microenvironment the aflatoxin. So that is the implication of this particular thing. So again, sorry, aflatoxin. Yeah. So the next one is the ochratoxin. These are again the chemical structures of this one. So these are types. This is occurrence. This is all about because of the aspergillus species, right? And some penicillium species. They produces this kind of systems. Again, so in this case, in the earlier case, we have seen the antibodies which was sitting onto the sensor surface. This is again a schematic representation I mentioned earlier also. So we have the scheme basically. So I'll take some water. Just a second.
Voice is not audible.
No, I am not speaking actually. I went to take a water.
Okay.
All right. So I'm back again. All right. Okay. All right. So in the previous case, we have seen that one of the receptor was antibody in the previous example. If I'll just go back quickly, this one, you can see this is the antibody. Okay. However, in the case of okra toxin, this is a little different receptor. That's the aptamer. So aptamer is nothing. It's a nucleotide sequence, and if I dissect the word, that's aptus. It means to hold. It means something is coming. For example, this is a molecule. When it comes, it folds. So that is the meaning. So antibody is not a foldable structure. If it will fold, it will denature. However, the aptamer is a nucleotide. If you fold it, I mean, you don't fold. The molecule folds. The target molecule folds it. So basically, what happens? We called it as what? Target induced conformational change. TICC, target induced conformational change. If it is the structure like this onto the chip, when the molecule comes, it makes some structure changes and the molecule sits here. Right. So, yeah. So now what is happening? This is the small chip. This is the surface. Okay. So this surface is activated by hydroxyl group, and this is called as a silane chemistry. This is aptes. Okay. This is one of the chemical. It's very common chemical. Now what is happening? What it is doing basically? It is providing the functional group. This one, right? And then this is what? This is the aptamer, right? This part, and this has a COOH, the carboxylic acid. So what will happen? CONH bond will form. What we can see here. So now this surface contains a receptor. Now just mentioning one thing very important. This M ball is nothing. It's a methylene blue. It's a dye, and at a particular potential, right? It oxidizes, right? So what is the point? Can everybody can see my hand? Just say yes or no.
Yes.
Yes, sir. Yes,
So this is the situation. That's the situation. When the molecule is coming, it is bending back. So the distance from this to this surface is different when it is not bent. If it is straight, the position from top to bottom is different. When it folds, the position from top to bottom is different, right? So that is the basic thing here. Now you can see here. Now what is happening? You see this folded here, right? Here. Now, sorry, on this side it's coming. So this electron, see, it is not moving to the surface. When it is not moving to the surface, the current will not generate. Now what is happening? This is what is the toxin, agrotoxin. So when it come into the system, it folded the way I explained here. It folded. Now this methylene blue, it oxidizes, and this electron will go into the into the circuit, right? When it will go into the circuit, and then the current will generate. So this might, I'm not might, this should have a concentration. So what you will get? The concentration on the x-axis. On the y-axis you will get a current. You keep on increasing the concentration of the ochratoxin, and the and the current on the y-axis will be keep on increasing. So that is what I mentioned here. It is explained onto this particular diagrams. So I'll not go into the detail of what is the meaning of DPV, or this is screen printed carbon electrode, this is what we are making. So again, we have to characterize again. मैंने पहले ही बोला था आप लोगों को characterize करना ही करना है. Somebody asked me in between, I guess Ajit, how we will know there is a purity or impurity. This is an EDX analysis. You can see here, right? By this particular analysis, you will know that how what is the concentration of silicon, oxygen, carbon, and so on. Whatever the materials are sitting there. So this is basically the same image, right? And then this is the situation. EDX spectra, as I mentioned here. This is another technique, what I mentioned already. This is the Nyquist plot. So this is a little complex system. This is EIS, electrochemical impedance spectroscopy. So anyway, so you can see this see on the y-axis and axis, that's just omega resistance value. As I mentioned earlier, you have a surface, you keep on adding something, the resistance value is increasing. You can see 20,000 omega, 40,000, 60,000, 80,000. You can keep on increasing the values. So after this process, what is happening? This is the calibration system. You keep on increasing the concentration here, and the current is keep on increasing. Why it is keep on increasing? Because of this reaction. So there was no current, but when you have included the ochratoxin here, the methylene blue is coming into the vicinity of the electrode and it is oxidized, and then you increase the concentration from A to H, from here to here. There is a linear correlation. So that's a concentration and that's a current, or nothing else. It's a signal. Basically it's a signal. So basically the signal is increasing with increasing in the concentration. Again, you can compare this system with the ELISA also, where on the y-axis there was OD, or somebody called as absorbance. So but here there is a current. That's the only difference. Otherwise the system is same. So anyway, so there is another example where we will be able to detect the herbicide. Again, this is a reduced graphene oxide, gold, this CHOX. This is one of the enzyme. Then you have to do a lot of the fabrication using the nanostructure materials, and then eventually when you're adding this one, you will get a signal output. So I'll not go into detail of every example, but there is a possibility that herbicide, every other molecule can be tested by using the nano sensors. So I'll skip this. Now I'll give you one more very interesting example which has been published recently. This is a leaf. Basically this is infected leaf, right? So why it has been infected? So there is a bacteria, sorry, there is a pseudomonas. There is a bacteria, gram negative bacteria. This bacteria can be detected. So these are the general information related to this one. So this infection can be monitored by detecting this particular bacteria into the leaf system. How to do that? So the current method developed is basically focused more on the molecular level. However, this is more extensive methods. However, for quick screening methods, the sensor can come into the play. Now you can see here that's a gold. It's not a particles. That is basically the electrodes. I'm sure everybody has seen a glucometer in the market. I'm sure about that, or maybe they have heard about that. So they have a small chips. So that is something like an electrode. Basically, that's a gold one. You can put some chemicals here onto that, right? Keep on adding that, and then this is antibody again for this particular bacteria. What I mentioned previously, the pseudomonas one, right? Now what is happening? This bacteria will bind onto the surface here, and then there will be reaction. This is a biological event. This one, this is the bacteria, this is the what, the system which is going to detect this particular bacteria. And these are the chemicals what people are using, glutaraldehyde, or this 4-amino thiophenol. These are the chemicals what people are doing. There are lot many chemicals. We cannot list one or two. Several thousands of chemical can do these jobs. So what is happening? Again, you can please see that this is little interesting diagram. I'll explain you. So this is the gold. See, axis is current. That's a potential. Gold, there is a current value. This, when you have added this chemical, you can keep on adding the chemicals. GA, glutaraldehyde. This is antibody. This is BSA. अब ये जितनी चीजें हैं यहाँ पे, for example, these are non-conducting. Current सबका drop हो गया, right? Oxidation current and reduction current. Both current is getting linear, but this is very high. This and this, because of the gold is a conducting surface. But every material what we have keep on loading onto the surface, it is reducing the electron transfer. Similarly, complementary to this one, this is the tail of a, sorry, that's a head, for example, and that's a tail of the coin. Another side of that. So this is the in terms of current, and this is in terms of resistance. You see, when you have added, this is for gold, this one. You see, you have added ATP, the resistance increases. You have added the GA, it is increased. You have added the antibody is increased. You have added the BSA mobile serum albumin, it is increasing. Resistance बढ़ रहा है, इसका मतलब current कम हो रही है. So these are the techniques. What if somebody is around Varanasi, they can come to my lab, you can see that, we can show you these things. So anyway, so you keep on increasing this one for some time, optimization, and then eventually these are the concentration in CFU per ml, that bacteria gram negative. When you are increasing the concentration of the bacteria in CFU per ml, you can see it is keep on increasing. इसका मतलब क्या है? What is the meaning? The meaning is what? Bacteria is insulating material. When it is insulating, it is not allowing the electron to pass the surface. That is why this is increasing. You see, this is what? Resistance. Resistance बढ़ता जा रहा है आपका on the y-axis. So after this whole process, we'll have lot more calculation, so that you can understand that what is the exact value and how it is selected. Anyway, so this is another example. You can see the similar bacteria here, Pseudomonas, by at the level of DNA. That was basically the direct bacteria in CFU per ml, but you can go for the detection of the DNA level also. There is nothing. This is the gold nanoparticles here. This is the real image. When you are adding the DNA of this particular bacteria, there is a shift basically before hybridization and after hybridization. Now how it looks like, this is the single stranded DNA, but when you are putting the gene of that, it will hybridize. When it hybridizes, there will be a shift, red shift this side, and then when you keep on increasing, this shift will be keep on increasing like this. And then you can make the calibration, the concentration of the DNA and the absorbance. So this is the output data. I mean, this is not in detail. I'm just putting the schematic of that. Again, in this case, there is a fusarium detection. In this case, you can go for there is a more technique. It is called an isothermal DNA amplification. This is a very common molecular biology technique. So you can also go for the detection using the optics by using the fusarium infected leaves, basically. All right. Now this is very much interesting. These are the commercial biosensor for several plant disease. A to C, 1 to C. This is for Phytophthora, Erwinia, and potato virus detection. And the important thing is what? This is marketed by these particular companies, right? Okay, so this is like a cascade. Everybody know that is a pregnancy strip in the market. So this is something like that. You can put some sample. You can take a leaf, pre-process it, sonicate it, put the sample like a urine drop here in the cascade, and you will get something like a line. So as I mentioned in my initial slides also, there is a lot of scope of paper-based sensor. This is nothing like, you can see this. Somebody is holding it in hand, this cascade, just dropping the plant extract, and then you will know that what is the level of that particular virus or Phytophthora or Erwinia, particularly these pathogens. So another is what? That's a ELISA kit for detection of citrus virus. So these are things which people can decode basically, right? And important thing is what? This is a hybrid smartphone based system, microfluidics device, which can identify the plant pathogen in a minutes. Microfluidic system is very tiny device where the fluid flows, and then it will be able to give the readouts. So that is linked with the smartphone. So Gene Z basically it is called. So these are the commercial devices in agriculture field, and we are expert basically in this type of devices in my lab. This one. Okay, so this is another type of a groundnut bud necrotic virus. This one, you can see the leaves. These are the infections in this thing. This is the importance of the general information of these pathogens, and these are the physiological roles. Now I'll tell you how the sensor is going to solve this problem. This is the most recent paper in July 14, 2021, most recent. Intertalenta is a very outstanding journal, very high impact. Now, how to make a sensor for this particular situation? In the plant system, you take a ITO sheet, that's an indium tin oxide, and then you spin coat like a spinning something. There is a method, spin coating. You can coat the graphene oxide, that's a GO is a graphene oxide, onto the surface like this, and you do some chemical steps in between. It's not mentioned in the scheme. So guys, this is the scheme basically I mentioned earlier in my starting slide. That's the schematics. So that's a hypothetical thing, and you have to do every step experimentally and characterize it. Then you will put in antibodies onto the surface. Now what will happen? You put, after putting the antibody, what is there? This groundnut bud necrosis orthotospo virus. This one antigen. I'll just call it the GBNV antigen. When it makes a complexation here, then you can study the response. So from where this antigen is coming, from the infected plant extract, just drop it onto the surface of this chip sensor, and then you will get a readout signal. That's an electrochemical signal, as I mentioned earlier also. So this is the fabrication from where you can extract directly the virus. Otherwise, you can go with ELISA or any other molecular biology technique. You can just complement the data again and again. So how to characterize? Again, as I said in the previous slide, आपने graphene oxide बनाया, you have to go with some characterization. That's a IR, that's a XRD pattern, and here again, just check it. This is the graphene oxide and ITO. That's a black line. When you have added something onto the surface, this is changing, and the blue is after changing this virus. So this reflect that something is happening onto the surface. After this characterization, now you see virus आपका non-conducting material, as I mentioned earlier also, the protein, the virus, the bacteria, and so on. So आप इसको increase करते जा रहे हो, the concentration from this to 150 nanogram per ml, the current is keep on dropping. There is a negative correlation. Generally, we see the curve like this, but you can see the curve like this also. So that's a negative correlation. So in this case, the signal, the concentration. So this is y is equal to mx plus c. This is the regression equation for this particular one. That's R square value. The sensitivity is 2 to 1 microampere unit gram and microliter. This one milliliter, and a linear range is 0.5 nanogram to 150. As you can see here, 0.5 to 150, and the LOD is 5.7 nanogram. This one. So that has been tested in tomato, cow pea, and this particular system, this particular virus. They have calculated the peak current in this case, and the comparison has been done with the ELISA also, as I mentioned. So you can do this virus detection using the sensor. That's a sensor basically, where you have used ITO, graphene oxide, this antibody. Similar antibody has been utilized for the ELISA. Now what's the difference? The difference in this case, ELISA, you need to have a plate reader. In this case, you no need to have a plate reader. You can just do the analysis in the small size mobile phone, even half of the size of the small mobile phone. So you can do that basically, right? Yeah, and it is super fast. Okay, so that's what you can do in this case. Now there is one more. I'm almost closing my presentation. Maybe I'll take 10 more minutes, then we can open for the discussion as long as possible. So this is another hormone, cytokine CKS. So this is the physiological role of this particular hormone. So now what is happening? There is a cytokine receptor AHK4. So this has ability to have the interactions, and in particular isopentylene adenine. This is one of the very important cytokines. So people have developed the sensor for this particular kind of the plant hormones. Now you see the fabrication again. That's a gold again. It's a cross-linking with the glutaraldehyde. There is a matrix generation, and then this is one chemical, N-hydroxyl acetonamide, succinamide, and this is another chemical which is help in the bioconjugation reaction. So this is ferrocene carboxylic acid. It will link here, and this will generate the signal basically. This reaction, carboxylic, this reaction will happen as I mentioned at a particular potential. This will oxidize or reduce. Now what is happening? You put this 2-IP isopentyladenine, sorry, and then there will be reaction. So this reaction can be monitored. So that is a basic fabrication strategy of this kind of a sensor, which has been utilized for the cytokine plant-based hormone detection system. Sorry, just hello. अभी चल रहा है, अभी टॉक कर रहा है. Oh, sorry. So this is the system by which you can have a detection of this kind of the hormones. Now again, what is happening? You are increasing the concentration from 1 to 11, just number. And then what is the number from this? These are the numbers, right? So the signal is keep on decreasing. So that is what I'm trying to explain. You keep on increasing the concentration of the material which is insulating, the y-axis current values will be decreasing, by which you can make a calibration system, something like this, with the concentration and the current value. Okay. Now this is the sample analysis and the root of the bean sprouts. You have added this concentration before adding, and you have added this one. So expected is what? 50 plus 107. It should be 157. But exactly the concentration was obtained 162. What is the meaning? That the 5 nanomoles were present in the sprout bean. This particular, because you have added 5 but you are getting 10. It means 5 is already present in the matrix. In this case, particularly root of bean sprouts. So the recovery percentage is basically more than 100. That is the reason. So again here you are expecting something this, but you are getting higher. You are expecting something this, but you are getting higher. So these high values means that plant hormone is sitting in this particular roots. So you can have this kind of the analysis by using the sensor system. So overall conclusion in the vision, what this is my last slide almost. So what is the overall conclusion is what? The plant pathogen hormone toxin can be tracked with the miniaturized sensing systems. So nanotechnology has the ability to revolutionize the agroeconomical system, and hence a new material can be engineered. That's why I mentioned about the nano engineering. The performance of the sensor depends upon two things. One is the receptor system, could be the antibody, how fast it binds, how what is the binding affinity, aptamers, enzymes. Another is the nanostructure material. You can see the nanostructure material can improvise the signal by several folds. I mentioned one of the example also. The signal is higher. So these kind of things can be mentioned. So this can be controlled in various manners basically, the nanosensors, and it could be suitable for agriculture by increasing the crop productivity. So now the ultimate thing is what is the integrated devices can have more potential implication in the agriculture field, and definitely the aim is what to have a wide dynamic range. For example, if you talk about the some level of compound in ppm level in the plant soil, if it is very narrow, the detection range is very shallow. But if it is very wide, the concentration can be tracked here, 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 everywhere. So that's the meaning of basically this term. So this is the overall conclusion, right? So finally, I would like to acknowledge my team: this is Vinish, Rohini, Deepika, myself, Rahul, Divya, Supratim, and all the students. These are my current group. All members are not sitting. I have 9 PhD students at this moment and several MTech and BTech students. So thank you very much, everybody. So if you have any further question related to my work or this one, you can browse my website chandraslab.com, and thank you very much.
Thank you so much, sir, for this informative talk. You explained every matter in a very elaborative and such a nice way, and I think this would help to motivate our researchers to work in the field of nanobiosensor. And now the session is open for the queries. If you have any queries, you can ask, please. If anyone having any queries, please ask.
Okay, meanwhile, I would take this opportunity to thank Dr. Chandra for giving us this opportunity and accepting our invitation on short notice and delivering such an informative talk here on this platform. And I must say that you have explained the concept of biosensors from the very basic, and every participant who is interested in this field, and we are having the participant from all different backgrounds. They are electrical engineers, mechanical engineer, as I was mentioning in the morning session also. So they would find something very relevant here in this presentation for them, and yeah, it is open for discussion, and we would love to listen from the participant, like if they have any query. Okay, since
Hello, sir.
Ajit sir, yes.
Sir, I'm Ajit Kumar here. Sir, I have a doubt. The virus detection is cost effective, or it's plus cost effective for planned virus detection.
No, it is not very difficult. Why I am saying it's a very cheap affair related if you compare with the ELISA system, because it's very miniaturized. So basically, what you need to just have a lot of wet chemistry steps in between to synthesize the nanostructures, layering and coding other things. Once you have optimized the system, then it is not that difficult. Moreover, you can have an
Okay,
on-site analysis. Of course, you need to
okay,
pluck the leaf and pre-process it.
okay, okay. Thank you, sir. Thank you.
Any other question? So if there is no question coming from participant side, so I must say thanks to Dr. Chandra again for this nice talk. Thank you, sir.
Yeah,
Thank
thank you
you
so
for
much
this session.
for calling
Yeah.
me for this very important meeting. And Arvind sir, thank you very much again, especially to you, because you have called me and you have remembered me for even last 20 years. Thank you again.
Thank you so much. Thank you. We'll meet again
Yeah,
sometime.
sure, sure.
And,
Yes.
yeah, we would love to have you here in this institute as well.
Yeah, definitely.
Yeah,
Whenever I am there, I'll just call you.
yeah, sure. Thank you so much.
All right. Yeah. Bye.
Sir, your mic is mute.
Okay, pardon for this. I was saying that it is very strange to see that the participants are entering very late in these meetings, and even the session is ended, some of the participants are asking to join the meeting. So, again, it is a humble request to all the participants to kindly join on time. I am going to give you a window of half an hour to join the meeting, because after that, I am going to restrict the entry in these sessions. Otherwise, what happens now that taking the attendance is very difficult because every time the numbers are actually increasing or decreasing in between the sessions. So this is one request. Second is that you all should leave the meeting. Otherwise, the time of overall time of the meeting is getting increased in many hours, because day and night the meeting is on if you don't leave the meeting. So you are all are requested to kindly leave the meeting as the session ends. So with this, we end this session today, and we'll see you tomorrow with a new session. Okay. Thank you so much for joining us today. Thank you.